Hello students, in our previous session we have come across and learned certain genetic terminologies. In our today's session we will continue to learn some more genetic terms in the chapter Inheritance and Variation. So let's get started. Parental generation. The plants used as parents in a cross are said to represent parental generation. They are designated by a letter P. Here, look at this figure here. There is a cross happening between a parent who is having purple flower and white flower. This particular generation is called as the parental generation and it is designated with the letter P. Filial generation. The progeny obtained as a result of crossing between parents is a hybrid progeny and is called filial generation. The first filial generation represented by F1. Here, the offspring produced as a result of the cross between the parental generation is called as the F1 generation. The progeny obtained as a result of self-fertilization among F1 plants represent the second filial generation represented as F2. Here, this particular generation which is obtained from the self-fertilization of the F1 hybrids. These are called as the F2 generation. Reciprocal crosses. A set of two reciprocal crosses means that the same two parents are used in two experiments in such a way that in one cross, one individual is used as female parent and the other as male parent. And in the second cross of the same genotype, sexes are reversed. For example, cross between a tall male and a dwarf female is reciprocal of a cross between a tall female and a dwarf male. Now, the purpose of a reciprocal cross is to find out whether both parents are making equal contribution. Look at this figure here. You can see in one of the crosses, you have we are looking at the shape of the seed here. We have the male having round seeds and the female having wrinkled seeds. And in the second half, we have the female having round seeds now and the male having wrinkled seeds. This is an example of a reciprocal cross. Back cross. It is a cross between F1 hybrid with one of its parent. Back cross is often used in genetics for the analysis of genetic constituents of the offspring. It's a quick way to make desirable homozygous individuals. Test cross. In this cross, the individuals of an unknown genotype is crossed with homozygous recessive parent. Please note, students, it's recessive parents only. In case of the back cross, the F1 hybrid can be crossed back to any of its parent. This cross is made in order to determine the genotype of the unknown individual. Now, why use only the recessive parent? Because in a test cross which is conducted to know whether an individual is homozygous or heterozygous for a dominant character, when you use the recessive parent, you observe the following results. The offspring will be 100% dominant if the individual is homozygous dominant, but the ratios will be 50% dominant and 50% recessive in case of hybrids or heterozygous individuals. Homologous chromosomes. The morphologically and structurally similar chromosomes present in a diploid cell are called as homologous chromosomes. Diploid cells, students, for your information, are the cells having the complete set of chromosomes. They are normally denoted with the symbol 2n, and haploid cells are the ones having half the number of chromosomes. Have a look at the figure here. This is a parental chromosome and this is a maternal chromosome. The paternal one and the maternal one. Here you have the same two sides of the chromosomes which are called as sister chromatids, and when the two chromosomes come together they form a tetrad. The They have identical gene loci positions bearing alleles. Here, have a look at the figure here. When the tetrad formation takes place from the paternal and maternal chromosomes, you will have the same locations coding for a particular character. That is, the genes are located at identical positions. Here, you have this location for the eye color, same at the paternal and the maternal chromosome. Same here is the locus for hair color in both the paternal and maternal chromosome. Students, something interesting here is the Punnett square or the checkerboard. The production of gametes by the parents, the formation of the zygote, the F1 and the F2 plants all can be understood from a diagram called as the Punnett square. It was developed by a British genetist, Reginald C. Punnett. It is a graphic representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and the left columns. All possible combinations are represented in boxes below in the squares which generate a square output form. Have a look here. This is a grid, and on one side you have the gametes, the possible gametes from the paternal side and the possible gametes from the maternal side on one of the columns. Then you simply bring them together, that is the capital A and the small a are brought together, and the grid is completed. Just have a look at another example here. You have along one side the father's allele, that is from the pollen grains. This is the possible gametes, and the mother's allele, that is the pistil. Here you have the possible gametes from the mother, and then they are simply brought together. Here two capital B's come together, one capital B, one small b coming together here. Then you have the capital B and the small b coming together here, and the two small b's coming together here. This is a diagrammatic representation of the possible alleles in a cross, which is called as the Punnett square. What does the Punnett square tell you? The boxes represent potential genotypes of offsprings if the two parents have offsprings. You can find ratios, percentages of the chance of an offspring will have the dominant or the recessive trait. Look at the grid here. Percentage of dominant trait that you can see. Dominant trait is represented by the capital letters. So in each of these grid you can find at least one of the capital letters. So the percentage of the dominant trait then becomes 75%. Ratio of dominant to recessive, it becomes 3:1. How to make a Punnett square? Form a grid first. From left to right, step by step construction of a Punnett square is shown in this particular diagram. The circles signify the gametes here. A stands for the dominant allele, and that's a capital A here, and you have the small a for the recessive allele at the same gene locus. Offspring's genotypes are indicated inside the square. So these here, the squares that you will fill will show you the offspring's genotype. So from you can start in this particular direction, or you can start bringing the two capital A's together, the capital A and the small a together. Either ways, it is going to help you just to fill the grid and complete your Punnett square. Things to remember while making a Punnett square. The Punnett square is a grid system for predicting possible genotypes of the offspring. So here you have your grid axis, each parents of the possible gametes. That is, you have your parents here and the possible gametes. Inside the grid, possible offspring genotypes, this which you will fill in bringing the two alleles together. The Punnett square yields a ratio of the potential genotypes and the phenotypes. So you draw the Punnett square with each row and column labeled with one of the possible gametes of the sperm and the egg respectively. So you have your empty grids, you have your possible gametes here. Fill in the genotype of the offsprings in each box by combining the male and the female gametes. So just combine the male and the female gametes. So in 3 steps, you have to remember while making your Punnett square is draw the Punnett square, write the genotypes of the parents and fill in the boxes. Punnett square activity or just a practice. Students, you can read through this entire instructions that are being given here. Pause this video for a while and try filling these grids. This will give you a practice of the explanation that I have given you earlier of formation of a Punnett square. Now, formation of learning this particular step is very important to understand Mendel's crosses. So make sure that you practice filling these Punnett squares without any mistake. Just bring these alleles together and fill in the grids. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care and ensure that you revise all the genetic terminologies that I have explained to you so that it will help you to understand the Mendel's crosses which I will take in my next sessions. Till then, be safe and keep learning. Thank you.